Hey guys, this is Kendall Terry. Once again, we've got, this is part 2 of cell communication. We're looking at different types of receptors, in essence. Here we're looking at receptor tyrosine kinases. These are abbreviated RTKs. There's only a couple kinases that will attach to certain amino acids. Tyrosine is one of them. Serine and threonine are the others. But tyrosine is the one that we see kind of most common in cells so far as for what we're looking at. We've got that they influence the cell cycle, cell migration, cell metabolism, cell proliferation. So there's a lot of things that RTKs are involved in. So let's look at actually what they do. How do they do this function in cells? So you have some type of ligand that comes in. These are the receptors, and you'll see that they're separated on the cell membrane. Whenever the ligands bind, the first thing that we have happen is a process called dimerization. In dimerization, those proteins will actually move in the cell membrane toward each other, and whenever they get close to each other, they will do a process called autophosphorylation. Literally, these tails hanging down will kind of fold up on each other, and whenever they fold up on each other, they will phosphorylate each other. So we say that they autophosphorylate, and they become little docking stations for them proteins to do different functions in the cell. This is obviously a generic version of that, so we're not looking at a particular one here. But if you see here, I've showed you a couple different ways that this can happen. So you've got phosphorylated proteins where a protein will bind or dock here at this protein and then phosphorylate an enzyme or some other protein that will then carry out a cellular response, or a protein will bind here to this docking station, be phosphorylated, and then cause a cellular response. There's various others that we could go into detail here, but for the sake of time and space, we're not going to really go into any more detail than that. When we're looking at signaling in cells, we've also got a couple others that we're going to talk about over the next couple of slides here. This one is called the MAP kinase. That stands for mitogen activated protein. Mitogen activated protein kinase, or MAP kinase, stimulates cell division by activating the normal pathways that control division. So we see this when a cell is needing to divide. So right before mitosis, you will actually see the MAP kinase proteins become active. Now, how they become active is through some kind of signal that's received by a receptor, and this part right here could be very different from cell to cell. It's not always the same process. But once there's an activator that started, from here down, we do see a similar process on this MAP kinase, where you start actually at the last one down here that actually deals with cell division, and every level you go up, they add another kinase to it to show you the process. So you actually start with MAP kinase, and it will phosphorylate a MAP kinase that will phosphorylate MAP kinase down here. So then you end up with these molecules that will stimulate the cell to divide. Now what you see here is what we call signal amplification, or a cascade effect, where you start one and then as it goes down it continues to get larger and larger and larger, and that allows the cell to then, you don't slowly work into cell division then. If the cell's supposed to divide, it sends a signal, and there's enough activators in there then to make that process work. That's called cell amplification. It gets bigger as it goes, as you can see. Now what we have found in some organisms like yeast, and we suspect that these proteins are also in others, although they're kind of hard to find, is that there are what we call scaffold proteins. A scaffold protein will actually have an attachment to each of the MAP kinase proteins needed, so that when this one is phosphorylated, it will immediately phosphorylate this one and phosphorylate this one, and then once this one is phosphorylated, it will give the cell response. So it'll hold them all together, so you get a boom, boom, boom effect and a response. This, though, will keep signal amplification from happening. So we know that some organisms like yeast definitely have these. We suspect that we see it in other organisms, but this protein is kind of a hard protein to figure out and find in research. The next one that we have is called the RAS protein. The RAS protein deals with a RTK receptor. So we've got the RTK receptor. I didn't draw this completely out. You can go back and look at what it looked like. and it will phosphorylate the RAS protein. Now, the RAS protein is very important to the cell because the RAS protein, a lot of times, is the protein that will initiate the MAP kinase cascade. So that is starting what we just looked at on the slide before this. But the RAS protein carries with it what we call a GDP molecule. That's a guanine diphosphate molecule instead of ATP. But it is an energy molecule just like ATP. So RAS will be turned on by RTK, making it a GTP molecule, which is the energy molecule, guanine triphosphate now. And that will then initiate the MAP kinase cascade. The important thing about RAS is that RAS can self-regulate and turn itself off. So it's turned on by RTK, and it can then turn itself off. So if you, let's say you're wanting to encourage cell growth, well, you might take a drug like a growth factor that would cause the cell to reproduce or divide. Well, right after that drug is given, it does initiate the MAP kinase cascade that can cause cell division, but that RAS protein can then turn itself off so that the cells don't continue to just divide. That could be bad. And in fact, what we have seen in cancer research is like 80% of cancer research is showing that there's a mutation that has occurred in the RAS protein, to where the RAS protein, once turned on, is not being turned back off. So you get reproduction, dividing of these cells, and a big clump of cells. Well, in essence, that's a tumor that develops from this because this protein was mutated. So this protein normally would be able to turn itself off so it doesn't continue to divide unless the RTK that responds to that one would turn it on. Very important protein in the life cycle of the cell. A couple others on here. I wish I could go into more detail, but just for this course and sequence, we don't really need to. But the Gproteincoupled receptors also have some different effects, some really cool effects in the cell. One is called cyclic AMP. Cyclic AMP is a second messenger. What that means is that once the ligand binds to the receptor, it will turn on a messenger that will carry that message then in the cell somewhere else. That is a second messenger. So from the initial receptor to something else that's going to take place, that's a second messenger that does that process. Cyclic AMP is one of those. It will turn on, it gets turned on by a Gproteincoupled receptor with multiple functions. And just to give you one, in muscles, cyclic AMP will cause PKA. You can look that actual name up if you'd like. It will cause PKA that breaks down glycogen to glucose, and then that also will prohibit glycogen formation. So in muscle cells, whenever you're working out, your muscles will use cyclic AMP to break down glycogen to make glucose, using cyclic AMP to turn PKA on. And PKA will also prohibit glycogen from being formed. So you get all these glucose molecules separated out so that they can be turned into ATP with cellular respiration. And it's not going to let any glucose start combining back into the glycogen formation. So it allows that muscle then to get the energy that it needs. That's just one area that cyclic AMP is used. There's various others. Another is inositol phosphate. This is also a second messenger, and it makes two very important molecules, DAG and IP3. DAG has multiple effects, just like cyclic AMP. We're not going to go into any detail on this right now. IP3 has a very interesting effect. When IP3 is created in this process, it will open calcium ion channels in the ER. So down in the ER, it's storing all those calcium ions in there so that they're not just running free in the cell. IP3 will bind to a ion channel. Diffusion will happen that will cause calcium ions to diffuse out. And whenever they're released into muscles, that causes the muscle to contract. So this is a very quick process that takes place. And all those calcium ions flooding into the muscle cell will allow that contraction to take place. But then it'll package all those calcium molecules back into the ER, and it will hold them there until IP3 is available to release them all again through diffusion to cause that muscle cell to contract once again. Very cool the way that works. And I once again encourage you to make sure you're looking at these in your book. There's more information than I could even go into detail here with. The last two slides deal with cell-to-cell interaction. This first one is tight junctions. In tight junctions, you have contact of the plasma membrane between adjacent cells, and it will actually weave them back and forth in sheets so that nothing can get through these cells. And we see this as an example in the cell linings of the digestive tract. You can see that on figure 9.17. But it will cause molecules not to be able to go between the cells. So your digestive tract, food's coming through here. You want all of that food to pass through a cell. You don't want it to go through between cells into your body cavity and blood supply and that sort of thing. You want it to go into a cell. So these tight junctions hold it so that nothing can get through there, so that everything that gets past this point right here has to go through a cell, and thus gives your body the ability to react to it and possibly attack it if it's a foreign invader, or so on and so forth. You've also got anchoring junctions. Anchoring junctions will mechanically attach to cytoskeleton. So you have two cells, and there'll actually be these junctions that will go from one cell through the cell membrane of another into its cytoskeleton. So you'll have these connection points back and forth, these rods, if you will, or even you might want to think of it as like a stretchy band, so that when one cell moves, the other pulls with it. Back and forth it goes because they're connected by these anchoring junctions. And you see this in muscle cells, obviously, to cause all of them to contract at one time. If you can get one moving, the others move with it. Also in your skin, that's what gives you that ability that your skin doesn't just pull apart. It's those anchoring junctions that help hold it all together. And those are cytoskeleton conjunctions there. This last one is communication junctions. And two that we're going to look at, gap junctions. That's passageways that permit small substances, such as sugar, to pass from one cell to the next. And what we've found in cell membranes is they'll have holes, and there'll be holes on the other one. And when those cells line up, there'll be a hole actually all the way through that cell membrane. And that way stuff can go from one cell to the other without having to go through the cell membrane. It saves time and energy for the cell. Things that need to go from one cell to the next, like glucose. But they can also, as the cell membrane moves, it can actually shift those holes to where they're closed off so that nothing can pass through unless it needs it for the cell. And that way it's regulated to a degree in the animal cell. In plant cells, you also have the cell membrane and a cell wall. So to get through the cell wall, there have to be holes in the cell wall. We have found that plant cells do have these. They're called plasmodesmata. So those are holes in the cell wall that allow the gap junctions in the plants to line up. And this connection then happens with the ER. Now there's a little difference there. The ER will actually go from one cell to the other instead of it just being an open hole like we see in gap junctions. So that covers cell-to-cell interactions and cell communication.